with you all. So today I'm going to talk about p53 isoform, but first I'm going to talk about a concept that will lead to paradox, and I will then lead to the isoform and try to complete some of the gap we have. So yes, I would like to engage with you if I have time. We'll see. So first is on the hallmark of cancer, so from Uganda, where you could actually split them into different organizations. You could organize them cancer cell autonomous part, so the component that make the cancer cells decide what it wants. You could also define the non-cell autonomous compartment, so that would be the tumor's trauma. And sometimes a bit difficult to split between the two, so some were on both sides. Okay? So cell autonomous and non-cell autonomous. And then I tried to write that as, I like mathematics, unfortunately. I tried to write that as an equation, saying that So the tumor behavior, so that will be the cell fate outcome, would depend on the parameters that define the cell autonomous component and the parameters that define the non-cell autonomous component. And I will link them by multiplications, because if there is no mutation, we will link cancer. And if the cells that surround the cancer cells do not let it grow, then they will be cancer. Okay, so I put an interplay between the two. So we know that p53 is very involved into the first category, the cell autonomous component of that pathway. There's plenty of publication in there. I put in red the one demonstrating that the isoforms are also involved in that compartment. And if you want a nice review to catch up with the isoform, if you don't know them, I will detail them a bit later, but that's a nice review that detail them. Then you have also the tumor compartment, and also here p53 is heavily involved in that aspect. There's some work, some Strohlab, Cameron, or Einstein. I put also in red here publication indicating that the isoforms are also involved in the non-autonomous, non-cell autonomous compartment. Okay. And then there is the multiplication aspect. So several publications have shown that actually p53, mutant p53, impact on the non-autonomous aspects. And reciprocally, the non-autonomous cell aspect, like the microbiome, will impact on the tumor, like Helicobacter pylori, or T cells compartment will do it. We have also the delta-133, for example, involved in regulating cell cells, and also the T cells being regulated also by delta-133, impacting that on the tumor. So it will mean that p53 pathway, I'm very careful. When I mean p53, I don't mean the single p53. I mean the pathway. Okay, broad, not single p53 protein. That's why I talk about p53 pathway. So the tumor behavior is proportional to the interaction between the autonomous cell compartment and the non-cell autonomous compartment. So, what is the weight of p53 in that equation? We know that p53 is not the center university or the universe, so it didn't much, doesn't go much. But yes, so p53 is not the only parameters within the cells. It is embedded in different cell signaling, and there is also heterogeneity. The tumor evolves with time. So what is its contribution to that equation, to the outcome? So we could test whether it's a heading bite or will be quite challenged by any other genes that are co-expressed with it. So if p53 has a limited contribution to tumor behavior, we would expect to see a low selection pressure during tumor evolution. Low frequency of tumors, we should expect low virus contacting it. We should, maybe people with germline mutation of p53 will be very affected by the tumor. The mutation wouldn't be very penetrant. And the critical point would be that restoring the right type TP53 gene alone would be sufficient to eliminate cancer in animal models. That is the killer experiment. So we would predict that p53 should not be among the most frequently altered genes in cancer if it had no impact on the food. Everybody knows that p53 is the most frequently mutated gene in cancer. So that defeats one aspect of that p53 is not involved, is not a key player in that. We know that for many patients, unfortunately, with only one single mutation in the coding sequence of p53, will develop cancer at high penetrance. We know also that p53 has been discovered through its interaction with viruses. And now we know plenty of viruses, and I'm not aware, doesn't mean it doesn't exist, but a lot of viruses, whether oncogenic or not, have developed strategies to target p53. Therefore, there is a strong evolutionary pressure to control the p53 pathway. The killer experiment now. Will it be possible if we restore the endogenous right type TP53 gene expression to drive tumor regression? And there are two key experiments published in Nature by Scott Lowe and Taro Jack in the lab, where they silence with an shRNA, the p53, in carcinoma, liver carcinoma experiment, and they switch off the shRNA. And by doing that, the cancer disappears. They restore one gene, p53. Despite all the mess that had occurred in the liver cancer, you restore wild type p53 gene expression, a precise endogenous TP53 gene expression, not a cDNA. You restore the wild type TP53 gene expression, and the cancer is gone. Okay? That's, for me, quite extraordinary. It really means that all the model assuming that he would have a minor role or he would be impeached by other genes, if you restore the TP53 gene expression, the endogenous TP53 gene expression with its entire influence, promoters, then the cancer is gone. So then the p53 paradox. So what we show is that p53 is the most frequently mutated gene in cancer. People with germline mutations develop cancer. We know that pathogens, virus, bacteria, target p53. And the most important experiment, you restore, and we had several talks during the conference showing that restoring wild-type TP53 gene expression induced tumor regression. Therefore, the p53 pathway is central. It is a critical determinant of tumor behavior. In that equation, having p53 playing on A, playing on E, and playing on the interplay between A and E, it is a factor that has an heavy weight on the decision. And yet, when we go into a clinic, we are unable to make use of that. Completely unable. So p53 paradox. We know that biologically central, but clinically not relevant, not predictive. Something is missing. It's really important to realize that. And mathematically, we should be able to predict. It's a key driver. I would say the driver. Why can't we predict? So what do we measure in the clinic? We measure p53 status and as a proxy, and we say it's mutated, it's inactive. Well, we know that not all mutations are equivalent and also cell type dependent, and will depend also on the context. Then we look at staining, particularly the full-length p53, looking at DO1, because the DO1 antibody detects the very end terminus of p53, and will not see other type of p53. By the way, DO1 detects a subset of p53, doesn't see all p53 protein. And also, if we look at p53 genes in nature, but knowing that we have p53 response elements in about every corner of the genome, we don't know if we pick up the right genes to understand what's going on. Anyway, those measurements are very poor to guide treatment or to guide diagnosis. It's too broad, too imprecise. What are we really measuring p53 functions? So one experiment that did Andreas Strasser was actually to knock out by a CRISPR in the exon 5 the gene, abolishing the full-length p53 mutant p53. What did he observe? That the cancer cells don't leave a dime. They still carry on. Okay? We don't have the mutant full-length p53, cells are happy. It doesn't bother them. What I would like to highlight to you is that he didn't knock out the isoform. There are still isoforms going on expressed. And the mutations are mostly in the exon 7 and 8. The most frequent one was one in the exon 5 here. So it means that all the isoforms carry the mutations. I will come back on all. But it means that the full length, if it's mutant, is probably not the one that runs the show. What he did, which is I found was also impressive, was to restore by a CRISPR in the mutant mutation to put the Y-type sequence. We would expect that those cells that have been mutated, accumulating mutation everywhere, probably even in the p53 gene, restoring only the point mutation that was going, that was wrong, would actually, you would not be able to restore the tumor suppressor activity of p53. But in fact, it could not observe, I know it's a negative result, it could not observe cells that survive when you restore Y-type TP53 gene expression. Despite all the mess that has those cells, only even within the p53 gene, or does it mean that the p53 gene has not been mutated? It's protected from the rest, I don't know. But, therefore, is canonical p53, the full length alpha, that I will call now TA-p53 alpha, is biologically and clinically relevant form of p53, enabling us to measure the function of p53, have been be able to fit with the math. I mean, everything is mathematical. It has to fit with the math. And we don't. It's incredible. So what was exactly restored in this experiment? What did Strasser, so here I'm putting that first publication that we did with David when I was his postdoc in his lab, where we demonstrate that the p53 isoform regulate p53 transcriptional activity, and we characterize all the isoform. We determine that there is an internal promoter in intron 4. We determine that there is a splicing in intron 2, that there is an alternative splicing in intron 9, leading to beta and gamma, and on top of that, the ribosome can define, not the ribosome, the cell will define through cell signaling where the ribosome will start to read the p53 mRNA. So you end up from one gene, you end up with 9 mRNA. I can tell you there is more than that, but for now I publish 9. And they all lead, they are all expressed in a tissue-dependent manner. What we didn't get with the equation I presented is that we could not predict, regardless of the tissue, regardless of the individual patient. What we include here, because p53 is expressed in every tissue, is ubiquitous, the full-length p53 protein, but not the isoform. The isoform are cell-type specific, organ specific. Therefore, they include a variability that we didn't get when we analyze only full-length p53. We develop antibodies, or I develop antibodies. I started with David and then I carry on. So here to show you where is DO1. DO1 detects the orange part here, the very end terminus of p53, meaning it doesn't see the others. So a measurement of p53 expression level, you were looking at a few percentage of the p53 protein, probably active one. We don't know. So the measurements were wrong based on that. If we look at expression level of a full length, it doesn't mean that it was associated with a particular response. I develop antibodies that detect in the DNA binding domain, and I use also some David in the DNA binding domain. Those antibodies so the DO1, so here we transfected each isoform 1/1 in H1299, and we tested the antibodies with it. As you can see, the DO1 see 2 or 3, and you need to run well to actually separate the alpha from the beta. If you don't have a 12% and you're going fast, you won't be able to separate them. Then an antibody made by Bourdon BP5310 that detects the alpha, the C terminus. Then the KGC that detects the beta. Then we did the KGC12 that detects all the isoforms through the DNA binding. Am I too long already? Really? Gosh. Okay. So, what I wanted to show is that, yes, the mutations are included within the isoform. But when you analyze your mutation, you need to be aware where is the mutation, because you will have a mixed population. If the mutations are in the N terminus, it will mean your isoform will be Y type, and you will have a mutated p53 in the N terminus, and 133 the same in the C terminus. Therefore, you need to consider that. How do they matter? So we know that if you stress with drugs, you're going to change. So you're in cells with mutant p53, you're going to apply different anti-cancer drugs, and you're going to change the expression of the p53 isoform. And if you deplete the isoform without depleting the mutant, then the cell will respond differently. Therefore, they are active. We validated that after UV treatment. I wanted to show you that slide because we have a problem to copy that, so I hope now we will be able to copy this. So that's the HTT116++ and P53 minus. Fogerstein lab deleted the exon 2. So therefore, when you blot with the O1, you don't see any P53 in the HTT minus because exon 2 presents the epitope of the D1. However, if you use a siRNA depleting all the different isoforms, you will see that the HTT minus express all the P53 isoforms. Are they active? If we don't have full-length P53, yes. They don't need full-length P53 to work. There are plenty of mechanisms possible, plenty to look for. So there is, and if you manipulate, you change the ratio of the isoform, you could change the expression of MDM2 or BAX or P21 and others. And you're going to change the response to UV. We have the same on this time a deletion in the exon 4, so we don't make delta 40 this time. We make only delta 133 isoform, and the same, I had a movie, I won't show the movie, that the depletion of the different isoforms change the behavior of the cells. We don't have any full length, and no delta 40. Okay? And when you stain, you will see them on DNA. So it means that the p53 pathway is still active in absence of canonical. The biological activity that we could attribute to the TP53 gene are real, but it is not full-length p53 alpha. It's not only p53 alpha. Okay? And in some cases, you won't have p53 alpha. And we validated by genetic that people born with a mutation in beta are actually cancer-prone. They are germline. The rest of the p53 gene is right type. They have only a single mutation in beta, and they are cancer-prone. Therefore, they are part of the p53 pathway. They don't have beta, or they have a longer beta, actually, in their case, they will be cancer-prone. And how it does that is by forming oligomers with p53 here. We could see that they modulate differentially the expression of the gene. We change the repertoire of genes. I think I'm going to finish there. How you could work, I'll give you a bit of hope, how you could work with that complexity. Instead to use one antibody, you will need to use several antibodies and do the ratio between the different isoforms to be able to understand how they interplay. Okay? That's one solution. And we manage that in multiple myeloma, where we could actually improve the prognosis. So that fits with the math this time. If we include the isoform, then we can answer that equation. You can solve that equation. And final slide is to show you that actually if you use isoform specific / post-translational modification by immunofluorescence, you will see different spots. And the analogy I do is that if you blood for H3 histone, you will see a big band everywhere, doesn't mean much. If you look for H3K4 and E3, then you will have a meaning. Okay? It's not the abundancy, it's the type of p53 that seems to enable us to solve the equation. Final one, next year we will organize the p53 isoform meeting in Trento, in 6th to the 9th of July 27. We never get money, so unfortunately, if we manage per sponsors, I'll be able to pay some travel, but I can't promise anything. I don't want to promise anything, so usually I invite, and then you come or you don't. And we adjust the program as a consequence. But if you want to learn how we manage to progress, it's good. Usually we let a lot of young people speak. And we choose the best, but we want to give them the opportunity to engage and to make an effort.